Good evening, one and all. On behalf of the Physiological Society of India, I extend a warm welcome to all of you. It's a pleasure to have you here for the 13th lecture in our ongoing series, thoughtfully curated by our educational committee. Over the past 12 months, we have had the privilege of hosting 12 engaging lectures delivered by eminent speakers, which covered almost all the systems within the realm of physiology. And now, as we transition into a new phase, going forward, we will focus on a specific topic in detail, which we hope would be useful to both undergraduates and postgraduates. Today's spotlight is on the resting membrane potential, a fundamental concept in cellular physiology. Our esteemed speaker for this session is none other than Dr. Oruganti Padmini, a name synonymous with expertise and passion in physiology.
Thank you.
Dr. Oruganti Padmini completed her MBBS from MKCG Medical College in Berhampur, Odisha. She pursued her DGO as an All India entrance candidate at IGMC Nagpur in 1993, where she excelled and became the university topper. She joined service as a gynecologist, putting her DGO qualification to practical use. Later, Dr. Padmini obtained her MD in physiology from Osmania Medical College, Hyderabad, Telangana in the year 2007. Her academic journey continued as she became an assistant professor in the Department of Physiology at Gandhi Medical College, Hyderabad. Currently, she holds the esteemed position of professor and head of the department in the Department of Physiology at Osmania Medical College, Telangana. Now, coming to her research and contributions, Dr. Padmini's dedication to research is evident through her publications. She has authored several papers in physiology and case reports in gynecology as well. Additionally, she has actively participated in continuing medical education events, sharing her expertise in both physiology and gynecology. It's inspiring to see someone with such a diverse background, ma'am, bridging clinical practice, teaching, and research. So grab your metaphorical lab coats, adjust your mental microscopes, and let's explore the intriguing world of resting membrane potential together. It's my privilege to welcome Dr. Oruganti Padmini onto the virtual dais. Ma'am, the stage is yours.
Thank you, Dr. Sujana. Good
Thank you,
evening,
ma'am.
one and all. I'm extremely grateful to the organizers to have me here today. And thank you, Dr. Sujana and Dr. Madhavi, who have helped me edit my PowerPoint presentation. Thank you very much.
It's our pleasure, ma'am. Madam, can you start the slides, madam?
Yeah, I need the slides.
Just
I
click
I
to
have to go
present
to
and
the
start
desktop.
sharing, ma'am.
Desktop. The desk.
Ma'am, did you click present over there, which is
Yeah,
on
yeah.
the
It's processing. Processing.
processing? It's okay, it'll take some time.
So the topic at hand today is membrane potentials or the resting membrane potential. So this resting membrane potential is the crux of the entire biological system. So it helps us to maintain our homeostatic mechanism, and it is the main part of the messengering system. So have you all noticed that when we touch a hot object with our fingertip, immediately the brain comes to know that it is a hot object and we have to remove the finger from that object. How is this messenger system executed in our body? The basic mechanism here is the entity called as the resting membrane potential. What is this resting membrane potential? This was recognized long back, almost in 1700, by Luigi Galvani, professor of anatomy, Italy, who discovered that when electrical stimulation was applied directly to the muscle or to the nerves leading to the muscle, frogs' legs vigorously contracted. So this indicates that there is some sort of messaging system which is propagated from the nerves to the muscle and commanding the muscle to contract. So what is this messaging system and what is the uh, basis for this and how the resting membrane potential is playing a role in this messenger system? And there were other scientists also, like Professor Bernstein, who enumerated or enlisted the factors that influence the membrane potentials. And as we all know, we come across Walter Nernst and Goldman, Hodgkin, and Katz, who have played a major role in explaining the membrane potentials and the value, and also in describing the electromotive force and other factors which are related to the membrane potentials. So, what is this resting membrane potential? So, resting membrane potential, as the name indicates, is the membrane potential across the cell membrane at rest. So, that is when the cell is in a non excited state. It is the electrical potential difference across the plasma membrane. So, there are cells in our body which are called as the excitable tissues. They are the neurons and the muscles. And when the resting membrane potential in these two tissues are particularly important because they play a major role in the messaging system, as I said, from the periphery to the center and from the center to the periphery. So, why and how does this happen? So, mainly the resting membrane potential is existing for signaling purpose. It is a sort of a transmembrane voltage, that is the potential difference across the cell membrane, and there is energy stored in this miniature battery which can drive a variety of transport processes. Rapid changes in these membrane potentials when the cell membrane is stimulated leads to the action potential, which are nothing but the impulses which are propagated from one point of the biological system to the other point of the biological system to execute the functions. So, if you take the examples, there are electrical phenomena, hormone secretion, and local changes in the membrane potentials which all execute certain cell functions. For example, take the case of heart. We all know that SA node generates the impulses, it is the pacemaker of the heart. But the impulses or the action potentials are propagated to the AV node, and finally, it is responsible for the mechanical systole and diastole of the heart. Similarly, if you take the skeletal muscle, the impulses coming from the spinal cord, they are propagated to the skeletal muscle fibers and command the muscle fibers to contract. So, membrane potentials are at work right now in your body and are always there until as long as you live. So, even in case of glandular cells, also the impulses which reach the glands, they command the glandular cells to secrete the hormones, and this is only possible because of the existence of the membrane potential. So, this resting membrane potential or the potential difference across the cell membrane at rest in a non excited state, how is it generated in the first place and how is it maintained? So, for this to be understood, we have to go a little bit deeper into the physics, because the biological system is so beautiful that it has adopted the mechanisms which are actually involved in the transport of the in the flow of the current, and also the battery, like because it is electrically polarized. So the cell membrane, as we can see here, uh, has positive charges on the extracellular surface and negative charges on the intracellular surface. So it acts as a cell battery or a dipole, having the energy which can be stimulated. And it also acts as a capacitor. Capacitor is, in physics language, we can say it is a two-layered insulated plate which separates the charges. So if you see the diagram here, the charges on the outside or the extracellular surface of the cell membrane are mostly comprising of sodium ions and chloride ions, and on the inside are mainly the potassium ions, bicarbonate ions, and the non-diffusible anions. Non-diffusible anions are the phosphates and the proteins. So they carry a negative charge. So we have to understand the principles of electrostatics thoroughly to understand the existence of the resting membrane potential, not only its existence, also when it is disturbed, how it is restored to the normal original value, and also when it is stimulated, how it goes on ahead to develop into an action potential and generates the impulses which are conducted to different parts of the body. So, there is a definite comparison between the wires, the cables in the physical system, and the electrical currents that are produced in our biological system. The only difference is the electrical currents in a metal wire are conducted by the flow of electrons, as we all know, and the electrical currents across the cell membranes in our body are carried by the major inorganic ions like sodium, potassium, chloride, calcium, and bicarbonate. Mainly the sodium, potassium, and chloride ions, and especially out of that, sodium and potassium are the most important ions which are involved in this maintaining the resting membrane potential and also restoring it back to the original state. These sodium, potassium, and chloride ions, what we were discussing, are carried through certain aqueous pores which are formed by the channel proteins. So these channel proteins, they are actually aqueous pores. They contain water, and these ions, they mainly are conducted through these channel proteins, and that is how the current flows into the inside the cell membrane. So coming to the ionic basis of the membrane potential. So here, as I already told you, the ion permeation through the membrane is important for the maintaining the resting membrane potential. It is basically called as electrodiffusion, because if you see this word, there are two things here: electro and diffusion. So if you take a non-charged particle, then you can simply mention diffusion because it moves down the concentration gradient from a higher concentration to a lower concentration. But when you are considering a charged particle, there is also an electrical gradient, and it will move from a high positive, either higher electrical gradient to electrical charge to a lower electrical charge. So there are two gradients here which are in process for the permeation of the ions. So that's why it is called as electrodiffusion. And when they are diffusing, they produce what is called as a diffusion potential. So diffusion potential is the value of the membrane potential or the resting membrane potential. So electrical current across the cell membranes, as I already told you, flow through aqueous pores. So there are three types of integral membrane proteins here, which carry the or which allow the ions to flow through. They are the ion channels, which open and close and permit the ions to flow through, electrogenic ion transporters, and electrogenic ion pumps. So in this diagram, if you see, there is a membrane which is separating the charges, and the potassium ions are inside the nerve fibers are more, and they are actually the sodium ions move from the outside to the inside, and potassium ions move from inside to the outside. Because the potassium ion number is more on the inside, it moves to the outside, and sodium ions are more on the outside, and they move to the inside. So what I want to tell you is there is a concentration gradient, and the ions are moving along the concentration gradient. So in the process, they actually establish a membrane potential or a potential difference here, as we shall see later. So the important concepts or principles for understanding the membrane potentials are, we have to remember certain points which govern this establishment of the membrane potential. So for a charged particle, a concentration gradient must first be set up. So if there are more positive charges on one side of the membrane and less positive charges on the other side of the membrane only, there will be a diffusion of the charged particles from the higher concentration to the lower concentration. That is one point. So initially there should be a concentration gradient for the movement of the ions. The second thing is there should be an electrical gradient. So electrical gradient is when there is a greater number of positive charges on one side, they will move to the side where there are lesser number of positive charges, but in the process they will be leaving behind negative charges. So a stage will come when the negative charges will pull back the positive charges, and they will not allow the positive charges to diffuse to the other side, even though remember there is a concentration gradient. So at one particular stage, the concentration gradient will be opposed by an electrical gradient, which is equal and opposite to that of the concentration gradient. And if it is a single ion on either side of the membrane, let us take potassium ion, then we say that the potassium ion has achieved its electrochemical equilibrium. The net movement now will be zero, even though there is a concentration gradient. So the electrical gradient will prevent the movement of any charge from one side to the other. So now the most important point here we should remember is, even though there is an electrochemical equilibrium, remember that there is a concentration gradient, but the net movement is zero because the electrical gradient negates it. So the value of the, now at this stage, when it has been fixed at a particular um, um, equilibrium, there is now a potential difference across the membrane. So this potential difference is called as the equilibrium potential for that particular charged solute or the charged ion. So for such a thing to occur, remember one important factor is mandatory, and that is a semi-permeable membrane. So this, if without a permeable membrane, this potential difference across a membrane cannot be achieved. And also permeability is important in the sense where it should be semi-permeable. That is, the ion which is most permeable through that particular membrane, the equilibrium potential achieved by that ion will contribute majorly to the membrane potential when there are multiple ions involved in the development of the membrane potential. So permeability is the capability of ions to flow across the membrane. Overall membrane potential will be closer to the equilibrium potential of the dominant ion. For example, at the resting state, the cell membrane is more permeable to potassium, as we shall see later. So the membrane potential at rest, or the resting membrane potential, is closer to the dominant potassium ion as compared to the sodium ions. So the potassium permeability of the cell membrane at rest is 4 times that of sodium ions. So hence the resting membrane potential in any tissue, let it be skeletal muscle or nerve, any tissue, it is much closer to the equilibrium potential of potassium ions, that is minus 90 millivolts in this example I have shown, than the sodium ions whose equilibrium potential is plus 65 millivolts. So now if you take the cell membrane at rest, so what happens? There is a potassium um, leaking out of the cell membrane. So as we all know, more potassium ions are in the intracellular segment compared to the extracellular fluid. So potassium ions keep leaking out through potassium leak channels. They are called as the tandem port domains. These are always open and permit an unregulated flow of ions down the concentration electrochemical gradient. The potassium channels are 100 times more permeable to potassium than to sodium. Remember, this particular phenomenon occurs at rest. So, potassium keeps leaking out to the extracellular fluid, and it is the work of the sodium potassium pump which restore the potassium ions the concentration to the intracellular segment, so that the membrane potential or the resting membrane potential is restored to its original value. So now I told you that the concentration gradient should be set up first. That is the most important thing for the membrane potential to exist. So how is the concentration gradient set up? It is done by two major processes. The most important one is Gibbs-Donnan equilibrium. So this was actually proposed by Willard Gibbs and Gibbs-Donnan in 1878. So they describe how the concentration gradient is set up in the first place. So they propose that in the presence of non-diffusible ions, the diffusible ions distribute themselves so that at equilibrium their concentration ratios are equal. So what does this mean? As I already told you, in the intracellular fluid, there are certain non-diffusible ions. They are the proteins and the phosphates which carry a negative charge. So in this example, if you see, the potassium and chloride have oriented themselves in such a way that there is neutrality on the X side as well as the Y side. But if you see, the potassium ions on the X side will be more because they have to also neutralize the protein ions which are present and which are not permeable through the membrane. So if you see if you multiply the potassium ions on the X side with the chloride ions, they will be equal to the potassium into chloride ions on the Y side. This is because of the presence of protein ions on the intracellular in the intracellular fluid, that is on the X side. So this shows you how potassium ions are more on the inside as compared to the outside. The second important process which is also contributing to the setting up of the concentration gradient is the sodium potassium pump. Though it contributes very less to the value of the resting membrane potential, it is most important for maintaining the resting membrane potential. So when there is any disturbance in the resting membrane potential, automatically it causes pumps out 3 sodium ions to the outside and 2 potassium ions to the inside, in the process maintaining the number of charges on the inside or the intracellular space and the extracellular space to its original state. So it is an active pump, continuously pumps, and it is an electrogenic pump because if you see the stoichiometry is the property of the pump. It is capable of pumping out 3 sodium ions out to the extracellular space, 2 potassium ions in the intracellular space, automatically causing more positive charges to accumulate on the outside, and therefore the positivity on the outside increases as compared to the inside. So because of its stoichiometry, it is acting as an electrogenic pump. So there is net deficit of positive ions on the inside. Though the potassium ions are more on the inside, the intracellular surface of the cell membrane is negative because of this pump. It contributes only -4 millivolts to the total resting membrane potential, but it is majorly important in restoring the membrane potential to its original state. So now all this discussion was revolving around a single ion. So when there is a single ion on either side of a semi-permeable membrane, we can easily explain the presence of one excessive ion on one side of the membrane. But in biological system and in reality, there are multiple ions which are taking part, and how do they manage to establish the membrane potential? Mostly the sodium and potassium ions, and very negligible role of chloride ions are there to contribute to the membrane potential. When multiple ions are there, the ions don't reach their electrochemical equilibrium. The electrochemical equilibrium will be in between, somewhere between the sodium and potassium ion electrochemical equilibrium. So they do not reach their actual equilibrium. And one more important point we have to keep in mind is, if you see the cell, that is the, this is the nerve fiber, and this is the extracellular space. If you see here, the entire charges which are present in the intracellular space and extracellular space do not take part in establishing the membrane potential. It is only few ions which are present on the surface of the cell membrane, intracellular surface or the extracellular surface, which are responsible for generating and maintaining the membrane potential. So actually, if you see deeply, the ion concentration gradient across the cell membrane, if you take into consideration the entire intracellular space and the entire extracellular space, does not change. It is only the surfaces of the cell membrane which carry the charges. And it is only the cell membrane which is acting as a battery, not the entire cell. So once we have understood about the resting membrane potential, its establishment and restoration, now how to describe the value of this resting membrane potential. So resting membrane potential is actually a driving force for the ions. So it is described in terms of millivolts, or electromotive force for the ions to move across the membrane. So traditionally, the electrical potential difference across a cell membrane is expressed by its value inside the cell relative to the extracellular environment. The extracellular environment is taken to be zero, and whatever value we are expressing is in the value which is present in the intracellular surface of the cell membrane in relation to the extracellular fluid. For example, skeletal muscle cells, cardiac cells, and neurons, they are between minus 60 to minus 90 millivolts. So this says that it is minus 90 intracellular surface potential, and the extracellular surface is zero. So the total will be minus 90 millivolts. That is the potential difference across the cell membrane. You can call it as a membrane potential or a potential difference across the cell membrane, or the resting membrane potential. It is, when you say it is minus 90 millivolts, it is minus 90 millivolts intracellular as compared to the extracellular fluid. Similarly, in smooth muscle cells and human erythrocytes also, there are basic resting membrane potential with different values. And an interesting point here is the different values are there depending upon the functions of the cells, how soon they have to be, or how fast they have to be stimulated, or how slow they have to be stimulated. All this also depends on the value of the membrane potential, and that is the reason why different tissues have different membrane potentials. So now to calculate the electromotive force of the membrane potential, or the driving force on the ion, single ion or multiple ions, we have to take into consideration a large number of factors, like the membrane should be permeable or semi-permeable, and there should be a constant electric field, and ions should move independently of each other. That is most important. They should not depend on the other ions, and a constant permeability coefficient should be there. And one more important point here is the membrane thickness. That is also most important, because when the ions are moving across a membrane, the membrane thickness, as we'll see later, they are important for the constant field equation. So the diffusion potential develops mostly depends on the electromotive force and it is measured in terms of millivolts. It again depends upon the polarity of the electrical charge of each ion, permeability of the membrane to each ion, and concentrations of the respective ions on the inside and outside of the membrane. This is most important because the greater the concentration gradient, the greater the driving force on the ion, and the greater will be the moment of the ion. The rate of ion movement through the membrane, how fast it moves, again depends upon the concentration gradient, the permeability coefficient for the ion, how fast it is permitted by the semi-permeable membrane to move, and it is independent of other ions. That is called as the independence principle. Thickness of the membrane, that is A, because the voltage difference varies linearly with the distance across the membrane. So the change in voltage with difference, the change in voltage with the difference, that is the electrical field is constant throughout the thickness of the membrane. This is called as the constant field assumption. So all these factors are important um, uh, when we are talking or discussing about the movement of the ions and their driving force, which are responsible for the movement of the ions. So here, if you take a single cell, the sodium and potassium ions on the inside and outside are in a particular ratio. That is, the sodium inside and outside is the ratio is 0.1 normally, and potassium inside and outside is 35. So depending upon these ratios, the diffusion of the ions, the electrodiffusion of the ions occurs, and the rapidity with which it occurs and the rapidity with which it is restored also is dependent on these ratios. So the points to remember are the voltage of the membrane potential is determined by mostly the sodium potassium chloride concentration gradients. Contribution to the voltage is proportional to the membrane permeability. The greater the permeability of the ion, the greater its equilibrium potential will contribute to the voltage. And electronegativity inside the membrane is caused by outward going of positive ion. So if one potassium ion, for example, moves out, there will be one negative space inside the intracellular space. The permeability of sodium and potassium channels undergo rapid changes. So, and this is important for the transmission of a nerve impulse. Permeability of chloride channels, as I said already, are very negligible. Mostly it is the sodium and potassium ions only which contribute to the establishment of the membrane potential. So the calculation of the diffusion potential of an ion. How much is the electromotive force or the driving force of an ion is there, that is which is measured in the millivolts, that is the electromotive force. It can be calculated for a single ion by the Nernst equation and for multiple ions by the Goldman-Cart's equation. So the equation here, which have been um, developed by the electrophysiologists, is Ex is equal to RT by Fz in potassium concentration outside by potassium concentration inside. So E is the equilibrium potential for potassium, Z is the valence of potassium, and potassium concentration outside is Ko and Ki is the inside the cell, R is the gas constant, T is the absolute temperature, F is the Faraday number. So all these put together will give us a value which is nothing but the electromotive force or the driving force on that particular ion when a particular concentration gradient is existing. So this particular 61.5 log is a value which is seen in the spinal motor neuron whose membrane potential is minus 70 millivolts. So the Nernst potential of an ion is described, which is the electromotive force which we have derived from the Nernst equation, is described as the potential level across a membrane that exactly opposes the net diffusion of the ion through the membrane. The magnitude is determined by the ratio of the concentration. The greater the concentration, the greater is the Nernst potential required to stop the movement of the ion. Nernst equation can be used to calculate the Nernst potential, And as we already know, the sign of the potential is positive if the ion diffusing from inside to outside is a negative ion. So in this example, which I have given, if the concentration of positive potassium ions on the inside is 10 times that on the outside, the log of 10 is 1. So the Nernst potential calculates to be minus 61 with inside the millivolts inside the membrane. So the Goldman-Hodgkin-Cox equation is an expansion of the Nernst equation where it takes into consideration all the multiple ions which are present in the on either side of the cell membrane, which is actually what is happening in the biological system. So the total electromotive force is the driving force which is applied to all the ions when they are trying to diffuse from one side of the membrane to the other side of the membrane. And this particular voltage of the membrane potential or the value of the membrane potential is also called as V rev. That is the reversal voltage, because at this point, if you just disturb this voltage, the entire current will be reversed. If this Vm is more negative. If it becomes more negative, the net driving force on a cation is inward. At voltages that are more positive, the net driving force is outward. So this is called as the reversal potential. So now that we have understood what is the electromotive force, the driving force, and which is existing in the form of the resting membrane potential or the membrane potential at rest, what are the contributions of different ions in terms of value? It is majorly, as I told you, the potassium ion which is contributing. So if you take any tissue as an example, for example, a skeletal muscle where the membrane potential is around minus 90 millivolts. About minus 94 millivolts is contributed by potassium ions, but it is negated by the sodium ions. So when you put these values in Goldman-Cox equation, the answer is minus 86 millivolts. So minus 86 millivolts is contributed by both potassium and sodium put together, and sodium potassium pump contributes about minus 4 millivolts. So if you add this together, it comes to minus 90 millivolts, which is the existing membrane potential in the cell membrane of a skeletal muscle fiber. So here an interesting point is the potassium ions equilibrium potential is minus 94, but the membrane potential is minus 90. So the potassium ions will always be trying to diffuse out in order to bring the equilibrium potential to minus 94. So if they diffuse out, the inside of the membrane will become negative. So that is the reason why at rest, potassium ions keep leaking out of the cell membrane. But if the membrane potential becomes minus 94 millivolts in a skeletal muscle fiber, it will be detrimental to the function. So what happens? The sodium potassium pump comes into play and pumps the potassium ions back into the cell membrane, intracellular space, to bring back the membrane potential to minus 90. So this is constantly happening in our body to restore the membrane potential. Finally, we come to the measuring of the membrane potential. How the membrane potential is measured. So here, this was actually developed long back. They used two electrodes, one an indifferent electrode which is placed on the surface of the membrane, cell membrane, and another one is a voltage sensing electrode which is a pipette filled with silver chloride solution, and it is impelled through the cell membrane. So these two microelectrodes are connected to a voltmeter and an oscilloscope for recording. So this shows the potential difference across the cell membrane. And then the pivot of the experimental basis for all the understanding we have about the resting membrane potential are two methods which are used to deduce the properties of the ion channels. One is the voltage clamp technique and the other one is the patch clamp technique. So these are mainly used for studying the drug channel interactions, receptor mediated processes, and biochemical regulatory mechanisms that take place at the inner or external surface of the cell membranes. Basically, these techniques will enable us to study the properties of ion channels, what is the value of the driving force of the ions through the ion channels, and how to deduce the properties in a small patch of the cell membrane where there are only a few ion channels, or in a large patch of cell membrane there are multiple ion channels. So basically, this voltage clamp technique was developed by Cole and Marmont in 1940s, and it is very interesting to note that in those days only they were able to discover these voltage clamp techniques and use them for studying the electrical signals across the tissues. And these techniques were used by Hodgkin and Huxley also, who won the Nobel Prize in Physiology or Medicine in 1963. And these are all very familiar names to us. So if we go deep into the voltage clamp technique, what is it and what is the principle behind it? So they have taken a xenopus egg, that is a frog's egg, and they have used two electrodes. One is the indifferent electrode, as I already told you, which is placed on the surface of the cell membrane. Another one is a voltage sensing electrode, which is impaled through the cell membrane. These two electrodes are connected to the voltmeter. So if you see here, if you see these graphs, we can inject certain current through this voltage through the voltage sensing electrode in order to disturb the resting membrane potential and convert it to a new value, and then we can observe how the cell membrane is reacting to this particular change in the voltage membrane by opening the channels or closing the protein channels, and in order to restore the membrane potential, in order to restore the membrane potential. So if you see this graph here, so if you change the membrane potential to a more negative value, then what happens? There is and clamp it. That is by injecting current on and off, keeping it at that particular value. Then they have observed that there is hyperpolarization and there is no flow of current. And if we move the membrane potential from its original value to a more positive value, by injecting current on and off, they have noticed that there are large number of sodium channels which open, and there is a diffusion of the sodium ions down the concentration gradient, increasing the membrane potential more towards the positive side, leading to a firing rate, and a runaway potential develops, which leads to the action potential. So lastly, we come to the patch clamp technique. It is a modified voltage clamp technique in which small patches are used to study the properties of the ion channels. So if there is this was developed by Bert and Neher, who also earned a Nobel Prize in Physiology or Medicine 1991. So if you see these diagrams, the first there is a electrode which is attached to the cell membrane, and a high resistance seal is produced. So this is called as the cell attached patch clamp, and here they have punctured the cell membrane so that the internal surface of the cell membrane can come in contact with the bath solution. Similarly, they have also pulled the cell membrane, and this is called as a cell attached patch. There is no disruption between the patch and the cell membrane. So here there are different varieties of these patches. That is where this is an outside patch. where the outside surface, the extracellular surface of the cell membrane is exposed to the bath solution. There is also an inside outside patch where the internal surface of the cell membrane is exposed to the bath solution. And in this process, they have changed the environment in which the cell membrane is placed in order to study the currents which are flowing through the protein channels. So all this led to a greater understanding of membrane potentials. And to summarize, the resting membrane potential is the basis of the homeostatic mechanism, the messengering system, and it has to be at a particular value in different tissues for them to function properly. And once it is stimulated or it gives rise to an action potential, it has got to be restored back to its original self or the value. So this has actually, we have all along studied the physiological basis. Now we have to correlate the clinical significance also. Suppose there is hypokalemia, decreased potassium ions in the extracellular fluid. Then what happens? There will be hyperpolarization, and the greater intensity of the stimulus has to be given to achieve an action potential. Similarly, there will be delayed ventricular repolarization also in the heart, leading to re-entrant arrhythmia. Suppose there is hyperkalemia, that is increased potassium ion concentration in the extracellular fluid. Then what happens? There is an increased refractory period because during repolarization, the potassium efflux will be decreased, and this inactivates sodium ion channels, and there are arrhythmias. So, for this reason, the potassium ions, the sodium ions, and other ions on either side of the cell membrane should be maintained at a particular value for all the processes to occur in the biological system, because major electrolyte abnormalities can lead to muscle spasms and seizures. So, uh, this explains how important resting membrane potential is in our body. Thank you.
Thank you, madam, for your informative lecture. It was very elaborative. I don't think there are any questions because it was so informative and elaborative. So now I would like to invite coordinator of Physiological Society of India Education Committee, Dr. Madhavi Lata, madam. Uh, she is a professor and HOD of the Department of Physiology in NRI Medical College for vote of thanks and further information.
Thank you. So good evening, everyone. So today's session was very enlightening and very, very important for the undergraduate students. And today's session is 13th episode of the lecture series conducted by the Physiological Society of India, that too the educational wing of the Committee of the Physiological Society of India. So I would like to extend my heartfelt thanks to everyone who contributed to its success. It has been an honor to be a part of this wonderful series for me. So on behalf of the Education Committee of PSI, I wanted to thank our esteemed speaker, Dr. Oruganti Padmini, who is a professor in the Government Medical College, Telangana, for her insightful presentation, very valuable knowledge that she shared for the undergraduate students mainly. But this is very important for postgraduates also. This is the basic physiological knowledge that is important. So, Padmini madam, your dedication and preparation are deeply appreciated, and you have made this event and this topic truly enlightening. Thank you very much. I also extend my thanks to our moderator, Dr. Sujana, who is assistant professor in NRA Medical College, and you are instrumental in making this presentation productive and meaningful, and your attention to detail and other efforts are very much appreciated, madam. Genuine and specific thanks to the audience for the time, interest, the interactions going on, which will keep this session live for a longer periods. So I also want to express my thanks to PSI Education Committee who are working behind the scenes, Dr. Vinay Chitturi, Dr. Arjun, Dr. Shempa. You are all helping us a lot in maintaining this whole process going on very smoothly. Thank you very much. So I would like to thank the Physiological Society of India very sincerely for their generous support, and particularly Dr. Prashun Priyanayak sir, who is president of PSI. He is a constant source of inspiration, lot of encouragement and help to us any time we ask for it. So thank you very much, sir. So finally, anyone I have forgotten, please excuse me. My heartfelt thanks for this program, which has run very smoothly. Thank you very much. So next program will be PSI Lecture Series 14. So this program will be conducted in the fourth Saturday of August, that is 24th of August. The topic is a continuation of today's lecture. This will be action potential, and the moderator, it is coordinated by Dr. Sabita Yogaraj, Professor, Government Medical College, Katwa, Jammu and Kashmir. So the speaker is Dr. Anjali Nadir Bhatt, who is Professor Physiology, JNU Medical College, Jaipur, Rajasthan. She is ex-principal of the GMC Katwa, Jammu and Kashmir. The moderator for the session will be Dr. Mehak Mukti, who is Associate Professor and Head of the Physiological Department in Government Medical College, Anantnag. Okay. So please join us on 24th August. So with this, we conclude our session today. So any doubts or questions? Yeah, it seems we have a few questions in the chat box. So, madam, please.
Yeah.
Ma'am, there is a question by Sheshav Somani.
Yeah.
He's asking to elaborate in detail on
Yes.
effect of hypokalemia and hyperkalemia on resting
Yeah.
membrane potential.
Yeah, yeah. If you take for example, first hyperkalemia, hyperkalemia means there are a greater number of potassium ions in the extracellular fluid, isn't it? So when the during repolarization of the action potential, the potassium ions efflux is there that happens by diffusion down the concentration gradient. So when the potassium ions are more on the outside, this efflux will be hindered. The concentration gradient is reduced. So there will be the next stimulus cannot come and stimulate the tissue. So repolarization will take a longer time. That happens in hyperkalemia. Whereas in hypokalemia, because the potassium ions are less in the extracellular fluid, what happens at here in this case the potassium ion efflux to the outside, that is the say extracellular fluid will be increased, so that also becomes detrimental. That is the reason why there is a so the tissue, the force of contraction, if you take the skeletal muscle, the force of contraction of the tissue will gradually reduce. So as that is the reason why there should not be hypokalemia and hyperkalemia.
There is another question from Dr. Sirisha Kalyani. So what is the difference between electrogenic ion transporter and electrogenic ion pumps?
Yeah, electrogenic. Electrogenic is the term when there is a driving force on the ion, so that if a positive ion goes to one side of the membrane, it becomes positive on that side, and the side from which the ion has been transported becomes negative. So automatically there is a difference in the charges. So there is an electrogenic change that is occurring in the membrane. That is the meaning of electrogenic. The difference between a transporter and pump is pumps use the energy to pump these ions, like sodium potassium pump, as we all know that. And whereas a transporter does not use energy. For example, if you take the tandem course, which I have discussed about the potassium ions, they are leaking to the outside down the concentration gradient, at the same time causing an electrogenic situation there. So when one potassium ion goes out, there is negativity on this side of the membrane. So this is the difference between transporter and pump. But basically, both are electrogenic.
Madam, there is another question by Prasanna Priya Nayak sir. He's asking to discuss the V reversal in
Yeah,
physiology.
yeah. Actually, the value of the membrane potential, which is at rest, that is the resting membrane potential itself, can be called as V rev, because if, for example, if you take a tissue where it is minus 90 millivolts, if you change the potential towards more negativity, then what happens if it becomes, let us say, minus 94 millivolts? This potential difference is in relation to the extracellular. We are talking about the intracellular. When it becomes minus 94 millivolts, what happens? There will be a movement of the positive ions to the inside to bring it back to minus 90. But if you are changing the voltage to a positive side, that is from 90, if you make it minus 85, suppose, for example, then there will be more negative ions either will enter the cell membrane, or more positive ions will go out. So in the process, what happens? There will be a total reversal of the current at this particular voltage. So it is called as V rev or V reversal.
So, okay, ma'am. Thank you very much. So there's a nice question and answer session. So thank you, everyone.
Thank you.
So we are. Thank you so much.
Thank you very much.